Hello. Thank you, ma'am. Uh, Hello, everyone. Good afternoon. I hope energy is still there for the last session. Yes or no? So, actually, myself, Srinivas, I am a first year research scholar, Department of Pharmaceutical Chemistry, Faculty of Pharmacy. So, today I'll be discussing about molecular dynamic simulation and its result analysis. Uh, so, first, we'll be having just brush up of previous sessions what we had. So we know central dogma, like from DNA, we'll be getting transcription, it becomes RNA, and then translation, it becomes protein. So from network pharmacology, like we'll be identifying the genes, which is important, which uh, develops the disease. So identifying the targets is crucial in drug discovery. So by using network pharmacology, we can identify the main target which is causing disease. So inhibiting or suppressing that target would suppress the disease. So that's the basic idea of network pharmacology. Then we had target selection. So protein, which plays a major role in the disease mechanism. So we all know, like if you take neglected disease, if you take cancer, protein plays a major role. So identifying a uh, protein which if we inhibit that protein, disease mechanism gets inhibited. If we identify the exact target, then of the drug discovery will be the easy. So then we had pharmacophore modeling. So a pharmacophore modeling is like selecting a mobile with good features. So if we have mobile with good features, so mobile will be running fast, good, everything. So drug which has good features will have good interaction and which can inhibit the disease. So that's the basic idea of pharmacophore modeling. Then we had AI and ML drug discovery. So ML is an extension of QSR. ML plays a crucial role in identifying a ligand which may show the action to a particular target. So generally QSR will be focusing only on one scaffold, but ML will be focusing on different scaffolds or different properties, which helps in giving the biological activity. So then we had molecular docking. Molecular docking. So molecular docking is one of the crucial step where in the drug discovery where we'll be identifying a drug which has interaction or which binds to the particular target. So it can be upregulated or downregulated. So docking gives a basic idea like whether it has interaction to a particular target or not. So by combining all this, now we'll be having molecular dynamic simulation where the both protein and ligand will be in a motion. So first we'll be seeing what is definition of molecular dynamics. So molecular dynamics here will be analyzing the dynamic nature of a system. So here both the atoms of protein and ligand will be in the constant motion. So identifying it will be in continuous dynamic in the motion. So here we'll be using computer simulation. So what is computer simulation? Till now we saw like we'll be seeing like in silico, in silico, in silico. So till now we saw like in silico, like what is that? So computer simulation, it involves mathematical modeling where we'll be using mathematical formulas to predict or to analyze the results. So which is designed to predict here in molecular dynamics, we'll be predicting the behavior of the complex like protein ligand complex. Here in the picture you can see protein in the docking, the protein will be in the static, ligand will be in the like conformation, like ligand will be changing its conformation and it will be binding into the binding pocket through different conformations. So in the dynamics we'll be seeing like both will be in the constant motion. So from real system to computer simulation. So here we have real system where we'll be in the labs, we'll be doing experiment, we'll be getting the results, we'll be analyzing that one. So some experiments will be there where we need a simulation, a computer simulation or experiment which we cannot do or which we have hypothesis, and based on the hypothesis we need a prediction. So that time we'll be developing a model. So computer model. So we'll develop a model, we'll be having a model. From real system, we'll be doing experiments, we'll be analyzing result. Same from the model, we'll be simulating, we'll be getting the model like simulation results. Or if we are not simulating, then we'll be having a hypothesis. For example, from yesterday's session, we are getting, so what is hypothesis? What is hypothesis? So hypothesis here, for example, if you are considering a cancer as an disease. So in cancer, we have like for drugs, we have resistance. To overcome the resistance, we need a new drug or we need to modify the drug. So that will be the hypothesis. That will be our agenda to perform the simulation. So we'll have a hypothesis. So we'll get a prediction from the like to apply the theory, we'll be predicting for the hypothesis, we'll be predicting. So comparing the real system results and the simulation results will increase the simulation results a greater way. So comparing real system results, simulation results to the hypothesis may increase our prediction more. So various computational approaches in computational modeling. So here in computational, we'll be analyzing two properties like structural and dynamical properties. In structural, majorly we'll be focusing on molecular mechanics. So it uses force field. So morning and now in the session, we'll be using, in the last session, we saw molecular docking. Molecular docking and molecular dynamics, it will be mainly working on molecular mechanics. So which uses force field. So in quantum mechanical, we will be focusing on the thermal properties, electronic properties of the particular structure. So that we'll be using quantum mechanical system. So in that we have different types, semi-empirical, ab initio methods. So as the list goes, the complexity of the computational increases. Requirements to perform MD. So for performing MD, we need a 3D structure or 3D like complex of protein ligand with the good connectivity, and choice of force field will be the main important criteria in the molecular dynamics simulation, because force field plays an important role in movement of atoms. So force field we need with good elemental coverage. Van der Waals radii bonding geometry, force constants, that all comes under parameters like bonding and non-bonding parameters that we'll be seeing in the next slides. So mathematical formula, like already I explained, like computer simulation, it will be going on based on the mathematical formulas. So we'll be using formulas to predict the result. Then for running molecular dynamics, we should divide dynamic simulation into different minimal steps, like few femtoseconds. If you divide, then simulation will be faster. Now we saw the definition. So we'll be having a query like why we need to do molecular dynamics. Till now we saw, generally in the papers we'll be seeing that they'll be doing molecular docking studies and they'll be getting the results. So to validate the docking scores, we need to perform molecular dynamic simulations. Here we can see we have a receptor, we have a ligand. So how ligand binds to the receptor or into the binding pocket, we can analyze through the molecular dynamic simulation. So in docking, generally what protein will be in the static, ligand will be moving, ligand will be binding into the binding pocket through different conformations. The conformation which has better optimal in the binding pocket and has desired interactions will be giving the good score. That generally the docking process will be going on. But in reality, both atoms of protein and ligand will be in the constant motion in our body. Like like proteins and the atoms in the MD, both will be in the continuous movement. So we have three types, like generally in computational, we have three types. Hello. So in molecular docking, will be like the receptor will be rigid, ligand will be the more like it will be flexible. So it is computationally not that expensive. The calculation will be faster. So we have another type of docking, that is induced field docking, that needs little bit more computational power. Why? Because protein will be not in the motion, only the binding site which we will be identifying that will be in the motion. So ligand and the binding pocket amino acids will be in the motion. So which increases our predictability and identifying the key interaction residues will increases. So here the flexible protein, in the sense the binding pocket amino acids will be flexible, the ligand will be flexible. So as the protein, the binding pocket is moving, so calculations will become, we need more computational power. And here we'll be seeing different conformations of interactions of amino acid with the ligand. So then comes molecular dynamics, where both protein and ligand will be in the constant motion, and it goes very slow when compared to induced speed docking, and it needs computationally more power than the docking. And here we can see different conformations of both protein and the ligand. So here another why we are doing molecular dynamics, in the sense we need to identify the stability of the drug binding to the receptor. For example, in the docking, we'll be seeing how it is binding, whether it has interactions, key interaction amino acid, whether it is having or not. Based on that, we'll be getting score. But during the molecular dynamic simulation, both it will be in the constant motion, so that we can get an idea whether it is throughout the simulation time, whether it is binding to the particular receptor or not, whether it is binding in the binding pocket or not, whether it is retaining the amino acid interaction throughout the simulation time or not. So that we'll be analyzing through molecular dynamic simulation. So differentiate ligands with similar docking score. Now, for example, we have here for ligands, for example, we have docking scores of 11, -11, -10.6, 10, 10.8, etc. If we have the scores with near values, that time we'll be generally opting molecular dynamics, because through the scores we cannot rely on the docking scores as it is. So by performing molecular dynamics, we can come to a conclusion that okay, among these ligands, this ligand with the highest, for example, -11, if it is stable throughout the simulation, if it has interaction throughout the simulation time, then we can consider that particular ligand is having a good docking score and a good stability. So in molecular dynamics, force field plays a major role. As I already told, force field, definition of force field as will be determining the potential energy of a particular complex or a system throughout the simulation time by using mathematical equation by combining the parameters. So here parameters are generally bonded and non-bonded parameters. So these are some of the force fields generally we'll be using. Charm and amber are majorly used in the freeware softwares, because the force field, charm and amber has more elemental coverage. In the 3D, like in the requirements for MD, I showed like force field with maximum elemental coverage. So amber, charm has maximum elemental coverage, and year by year they will be updating for the different elements. So considering this, you should select the force field which has maximum coverage. So charm and amber, as we have used, it has major elemental coverage. Both is developed by Schrodinger, which is a commercial force field that we need a commercial license to utilize it. So in the molecular dynamics, first we'll be analyzing the interactions. That there are two types: bonded interactions and non-bonded interactions. In bonded interactions, we have different types like bond stretching, bond bending, and torsion energy. So consider this as an hypothetical molecule. We have different bonds. So bond, the bond we consider here, it we'll predict this by using Hooke's law. So we'll be considering the bond, the atoms, two atoms as a ball, and the bond will be as a spring. So stretching and bending of the two balls will be analyzing the bond stretching by using this formula. No need to memorize this formula, no need to know about this formula. The software will be taking care, the software will be using the different, like from that software has a database. So from the database, it will be having the parameters. The parameters are Kr, constant, Kr constant, R equilibrium. These are the parameters which we'll be focusing. So the parameters, the database already has the parameters. So it will be calculating based on the atoms which are present in the complex of our protein and ligand. So from the bond, from that database, it will be calculating the bonded stretching energy. So then we have bond bending. When we have three atoms, there will be seeing the angle how when we bend the atom, so there will be a compression and relaxation. So change in the angle we can predict by using bond bending. So we have the formula for that also. So the database will be seeing the atoms which are present, and it will be calculating the bond bending energy. So then we have torsion energy, where if we have 4 atoms, so some part of the like the bond will be freely movable. It will be flipping that side in the other directions. So in torsion energy, when the two conformations generally we'll be seeing trans and cis. So we already know from intermediate chemistry that trans will be more stable than the cis. So based on these conformations also, it will be calculating the torsion energy. So we have that formula for that one. So parameters as already. So here I already told the constants and the other zone as the parameters. So how the parameters will be selecting based on the atoms which are present. For example, we will be having carbon, 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 nitrogen, carbon, oxygen, based on the different types of bonds and between the atoms. So we'll be considering that one. So based on the, for example, if we consider carbon and carbon, we'll be having different type. For example, hybridization will change sp3, sp2. It is aliphatic or aromatic. Based on different criteria, we'll be having the database. In the database, we will be having the values for particular parameters. So as I already told, force field, the force field should contain all that. It has a database. So it should have elemental coverage as the that parameters data. So while using different softwares, you'll be getting different results. Have you noticed that one? For example, during docking and during different computational tools which we use, we'll be getting different results. That's this the reason why we'll be getting different results, because it doesn't major softwares will not, majority softwares will cover. For example, commercial softwares like OPLS, they will cover entire elemental coverage up to 95%. And in free tools, for example, Amber and Charm, they also has elemental coverage. So this parameters plays that software plays a major role. So based on that, we'll be getting different results. So based the parameters, how they'll be calculating, in the sense they'll be using literature, they'll be using the experimental value they'll be using, or they'll be generally going for AB initio calculations, where they'll be getting the results for this particular parameters. Cross terms. So generally in the bonded interactions, just we saw an atom or 3 atoms or 4 atoms. So when we generally, if we stretch a molecule or if you bend a molecule, so opposite to that or connected to that atom has an effect. So that effect also we need to calculate. Generally we have stretching. So that stretching will be connected to the bending. For example, if you stretch, some angle will be bended in that. So that will be we need to calculate that also. If you calculate that also, your result will be more optimal. So we have different cross terms like stretching, bending, and bending. So combining all this like single parameters like bond stretch for only single atoms, or considering the effect of stretching to the bending, will increase your result accuracy. So we saw the bonded interactions. Now we'll be seeing non-bonded interactions. Non-bonded interactions also plays a major role in the molecular docking or binding of ligand to the receptors. So here we can see we have electrostatic interactions. For example, between nitrogen and oxygen, we have van der Waals of attraction and repulsion. We have hydrogen bond interactions. So in the docking, you will be seeing like you will be having this non-bonded interactions majorly. Pi stack. These are all interactions comes under non-bonded interactions. And we have Lennard-Jones potential, where it avoids coming close of atoms and have steric clashes. It calculates that one. So after calculating the interactions, now we'll be calculating the force experienced by the each atom. So for example, if you consider atom 1, 2, 3, the atom 1 will be experiencing the force from 2 and 3, and vice versa, like 2 experiencing from 1 and 3, 3 from 2 and 1. So generally we'll be calculating this force on each atom by using this simple linear algebra. As per Newton's second law, acceleration is dependent on force experienced by the object and mass of that object. So in the molecular dynamics, as we are using force field, the atoms will be in the constant motion. So we can calculate the acceleration by calculating the force experienced by the each atom. So from the acceleration, from the force, we'll be calculating the acceleration. From the acceleration, we'll be calculating the velocity. So from the velocity, we can calculate the position of each atom at respective time. So to summarize this one, generally in molecular dynamics steps, what happens? First, we'll be having a model. It will be having the atoms will be in the initial position. So we'll be calculating the force. We'll be calculating the acceleration. We'll be calculating the velocity, and then the atom moves to the different position. Here, for example, you can consider the black one as initial position and gray as it has moved to the another position. So from that equations which I have given, we can calculate that like position with respect to time. So generally this and all takes within just femtosecond. Generally we should carry this calculation under femtosecond, because as you increase the time scale, the computation, the simulation becomes more complex. So now we will see what steps generally involved in doing molecular dynamic simulation. First, we'll be preparing a molecule. For example, if you want to run for particular protein, only protein, not protein ligand complex, then we need to first we'll be downloading, we'll be preparing the molecule, or complex we'll be taking to the particular software, we'll be preparing that one. So after preparing the molecule, we'll be adding the box, that is periodic boundary condition. We'll be restricting the complex to that. We'll be solvating that one. We'll be ionizing that one. We'll be neutralizing that one. We'll be equilibrating that one in presence of ensembles. That is NVT and NPT are the ensembles. Then we'll be going for the simulation. Then we'll be analyzing the results or trajectories which we get. First, we'll be seeing like preparation of molecule. Generally, we know we'll be taking the molecule. We'll be if the hydrogens are missing, we'll be adding loop chains, side chains. If it is missing in the complex, we'll be adding that one. So that will be the general preparation of the molecule. In while doing the demo, we'll be showing how it works. So next will be we'll be adding the periodic boundary condition. So what is periodic boundary condition? We are restricting the movement of the atom in the particular box, or we are giving a restriction to the atoms. Yes, the atom should be in the particular place or in the particular box. So the central box or the box which we are seeing will be the main box, where atoms will be in the motion and it will be equal. So rest all, it will be a replica. Like if we consider a cubic, in all directions the box will be there. The movement will be from one box to other box. It will be continuous. It will be moving. So from this periodic boundary conditions, what we can understand, or what we'll be getting, is how much pressure and how much stress we need to apply to the particular atoms or particular boundary box. Because when we apply a particular pressure or stress, then the atoms will be moving. So from this we can estimate the density and the volume of the box and area of the box, like how much atoms is covering the entire box. Solvation. So generally the molecular dynamics, why we are doing, because it is mimicking our human conditions. We are mimicking that one. So adding solvent or missing addition of the solvent will be leading to the artifacts, or will be getting the wrong results. So generally in the molecular dynamics, what we can add water, we can add ions, we can add the lipids. For example, if you are targeting tuberculosis, where we can add membrane to the particular protein, we have that one also. So generally in solvation, we have two types: implicit solvent and explicit solvent. Implicit solvent, here you can see it is generally covering only the molecule, not entire the box. So the calculation will be faster, but the result will be not that accurate. Then we have explicit solvent. The explicit solvent, it covers entire the cell, as the PBC, the periodic boundary box, which in the previous one which you saw, we are restricting the complex or the particular atoms. So the explicit solvent covers entire the periodic box and the complex which we are restricting to that. So as we are using, we are covering the entire cell. So the calculation takes so much time, and the results will be more accurate than the implicit solvent. And we have so many water models like single point contact, tip 3P, 3-point contact, 4-point contact, 5-point contact. So as you see, the water models atoms also increasing. So if you use more like tip 4P, tip 5P, the simulation time also increases, the complexity also increases. Generally, for normal molecular dynamic simulations, we'll be using tip 3P. So which is computationally feasible, and we can get accurate results from that. Ionization. In the picture, you can see like the balls which are moving around the atoms are the ions which we have added in the molecular dynamic simulation. So here ionization, why we are doing, we are neutralizing the complex, which while adding the solvent will be having like dielectric effects by adding the solvent. So to collect electrostatic interactions, we saw the non-bonded parameters like van der Waals, everything. So to get accurately, we'll be ionizing the complex, because our body contains also ions like sodium, chloride, potassium, everything. So we'll be adding ions just to neutralize that one. So in ionization, we no need to calculate how much we need to add. Algorithms already they have been optimized to calculate. If we give a complex, it will calculate how much ionization we need to do or how much ions we need to add. Minimization and equilibration. So minimization in the software, what we'll be doing while the preparation of molecule only, we'll be doing the minimization. So in the steps, it is come at last, but minimization, what we'll be doing generally doing the preparation, we'll be adding extra like atoms or this one, hydrogens, everything. So there may be a steric clashes, or atoms may come closer, which may have steric clashes. So we'll be minimizing that one so that it attains the local energy minimum, and we'll be getting so the complex will be equilibrium throughout the simulation. So then we'll be doing equilibration. So while building the model, we'll be adding water, ions, everything. So while building, it may have some effects on the complex. So to neutralize that one, we'll be doing equilibration. Generally, while building, we'll be from the model building, we'll be having that artifacts like while doing. So to minimize that one, we'll be generally using NVT, NPT ensembles to maintain the equilibration throughout the simulation. So in equilibration, it uses statistical mechanics where we can understand the thermodynamical behavior of the particular complex throughout the simulation. For example, we have NVE, number of atoms, volume, energy, number of molecules, volume, temperature, number of molecules, pressure and temperature. So we have different types of ensembles in the softwares. So generally during the molecular dynamic simulations, we'll be using NPT ensemble because it resembles the lab conditions, and we can get accurate results by using this ensemble NPT. By using we can use NPT also, but in NVT what happens when we use NPT, there will be a sudden rise in the temperature and there will be no maintenance of equilibration throughout the simulation. So that's why as NPT it is close to the lab condition and it gives results which is near to our lab results. So that's why we will be using NPT throughout our simulation. So till now we saw how to prepare molecular dynamic simulation, what are the steps. Now we have a doubt like how much we need to run a molecular dynamic simulation. So on that picture we can see, based on your hypothesis, you can decide how much time scale you need to run. For example, if you are building a protein, like from homology modeling, only you are preparing a protein, there will be no docking or there will be no complex with the ligand. So generally from protein which we develop from homology modeling, we'll be seeing its stability throughout the simulation. For example, the protein folding, side chain folding, everything we'll be seeing. So for the particular protein ligand complex, generally we'll be going till nanoseconds. So till nanoseconds, we can see the bond stretching, the like bonded parameters and non-bonded parameters, and we can see the protein folding. And if side chains, if there is a continuous motion, so we can change that. We can optimize that complex, and we can run again the molecular dynamics simulation to get accurate results. So till now we saw how to develop a molecular dynamics model, everything. Now we'll be seeing different types of results which we can expect from molecular dynamics simulation. There will be like RMSD, like root mean square deviation, root mean square fluctuation, secondary structure analysis, covariance and PCA, hydrogen bond analysis. These are the different parameters throughout the simulation you can check from the complex. So now we'll be seeing the applications of MD. So we, as I already told, we have structural properties and we have dynamical properties. So from in the applications, we can see in the kinetic properties, we'll be seeing the structural properties of the particular complex or the protein. For example, if you have generated a protein, or if you have developed a protein from homology modeling, we can see, we can refine the protein, and we can refine the complex also. Then we can check the stability of the protein for particular simulation time, and we can check the protein folding, and we can check the protein ligand in stability and the transport of ions and ligands. That transport of ions and ligands we'll see in the next slide. We have the particular MD for that. So next we'll be seeing the thermodynamical properties. The MD has both applications, but generally for the particular protein and ligand, we'll be focusing mainly on kinetic properties. These thermodynamical properties, which will be focusing mainly on the atom-wise interactions, where we have different types like free energy perturbation, free energy binding, MMGBSA, MMPBSA, and heat of vaporization, pressure, boiling point. The first FEP, MMGBSA, MMPBSA, that will be analyzing the free energy after binding the ligand to the protein. So we'll be in MMPBSA or in FEP, we'll be calculating the Gibbs free energy or free energy after binding ligand to the particular protein, which is computationally more intensive and expensive. So here, as we are generally seeing the stability of the protein and ligand interactions, we can just focus on kinetic properties. Once we do docking and we need to validate the docking scores, for example, we have so many methods. In that, if you want to see the atom-wise interactions with the amino acids, we can focus on MMGBSA, MMPBSA, FEP. Specialized MD. So generally, molecular dynamics, till the procedure we saw, we have different types based on our hypothesis, based on the work which we want. We have simulated annealing, where we'll be doing with the different temperature, we'll be checking the complex with the use of thermostat to maintain the equilibrium. Then we have targeted MD or DMD, where in the last slide I showed in the kinetic parameters, where we will be seeing the transport of ions and this one. So if you consider a GPCR or ion, like the receptor, we can see the closing and opening of the particular receptor by using this targeted MD. So this all like it will be like an extension for the particular software. So if for commercial software, we can just run by a click. For a particular GROMAX, everything, we need to add like an extension for that one. So then we have steered MD. So in the steered MD, we'll be seeing the atom-wise stability of the particular ligand with the protein. So then we have replica MD, where we'll be combining with the Monte Carlo simulation, where we'll be seeing the extensive, if we want to develop a particular protein and if you want to check the stability of the protein, then we'll be using replica exchange MD, where we'll be seeing the conformation of the protein by using this one. And Langevin and Bromian dynamics, it is normal MD, which in all the softwares, it will be pre-installed and it will be calculating everything. So now we'll be seeing how to do a molecular dynamics simulation by using Desmond. Desmond is a software which is developed by Schrodinger and D.E. Shaw Research. So this Desmond is owned by Schrodinger, which is a commercial software, but for academic purpose, you can apply for license and you can take for that. And it is free, that is academic, which we can run in Linux, not in other OS. The free tools which generally we can use are Bromax, NAMD, VMD, are some of the software tools which are open source. Bromax is worldwide used free source software for the running molecular dynamic simulation freely. So that's like Bromax has some extensions, everything, but Desmond we can calculate only molecular dynamic simulation. So for running thermodynamical properties like FEP, everything, you need a license for commercial license for that. So here, generally, we'll be showing normal molecular dynamic simulation. Till now, any questions? Any? Come on, Mike.
So in docking, you've already prepared the protein and you have docked it with the ligands. The ones with the highest highest scores only you go and give it for dynamics.
Yes.
When we go to dynamics, why you have to again prepare the protein? You're using the same complex which protein is already prepared, right?
Okay.
You've added the side chains, loops.
Ma'am, for example, as I already told, different softwares has different force field. For example, Desmond has OPLS. We'll be using normally like open source. The force field elemental coverage will be different. So once you, for example, if you load into the Desmond, we'll be going for preparation because we need to minimize, we need to add like if any missing side chains or if hydrogens are missing, generally we'll be adding. We need to minimize the complex because during the simulation it should be equilibrium. It should be in the equilibrium throughout the simulation. That's why.
But if you prepared using glide.
Glide.
Glide is stored in the same.
That time we no need.
Then we don't.
No need. No. Generally, if we are using open source softwares, we need to we need to prepare and minimize in this for software also. Still, any other questions? Result analysis. Once we complete the demo, I'll be explaining again how different trajectories, how to see, analyze. So generally, these molecular dynamic simulations we cannot run in our normal laptop because the both complex like atoms will be in constant motion. It will be generating more heat and it need more computational power. So please don't try to run your molecular dynamics in your laptop. It may crash like anything. Any other questions, queries? Any other steps to be repeated? ಅಪ್ಡೇಟ್ ಮಾಡ್ತೀರಾ? So here we have our GPU, which we generally use to do molecular dynamics simulation. It has i9 Intel configuration with 12 GB graphic card with 1 TB SSD and 1 TB HDD and GeForce RTX 3090 Ti. So it has 10,800 CUDA cores. So CUDA cores, you can just you can imagine like one work will be divided for 10,000 members. So that is known as CUDA cores. So it has 10,800 CUDA cores. This is the GPU which we are using in our faculty, in our department. Based on your requirement and based on your complexity of simulation, you can select your GPUs.
Here is one online question. There are any other website where dynamics can be performed other than which you have?
There are open source softwares also. Now we are giving demo for Desmond, which is free for academic, and we have academic web servers. We don't have any web servers.
Yes, web servers are available. You can able to do with the Google Colab. Google Colab will be allowing you to perform GROMAX, not the Desmond one. So the Desmond one, you can able to perform only with the systems. There is no online cloud things under. Rather than that, I think if I'm not wrong, Galaxy is the server name where they are also offering the things. And other than that, WebGrow is a server which is offering to perform your molecular dynamics, but limited. 1 nanosecond, 5 nanosecond, less than that only they can give because it requires more of a hardware support. So that's the reason they're not offering like in a free, like a mini web server you can able to see to carry out your molecular docking, specificity like that and all they are giving free, right? But Desmond they could are not dynamics they can't do because it requires the high hardware support. Without high hardware support you can't able to do. That's the only reason they'll be doing. Hmm?
start. Huh? So we'll start the demo. So priorly, we need to install. Now we are showing the Desmond software. So which is we are running in the Linux operating system. So when you open the terminal, if you give Schrodinger/Mastro, you can open the software. Like we'll be running in the coding through in the Linux OS. So once you open, this will be the open like theme of this particular Desmond. So after once you open, so generally you should first you should create a working directory where all the results, all the particular files will be stored in the particular folder. So for to do that, you can go to the file. File. So you can change the working directory. If you already opened the software, you can change the working directory for the new folder, or you can create the working directory. From the working directory, in the working directory, you can save the protein ligand complex. So there are two types to open the complex. One is from the import structures, that is from file open file tab. File tab. File tab. File tab. File tab. So here you can see the import structures. So from the by clicking the import structures, you can import the complex, or by generally clicking the Control+I, you can import the structure. Import structure. The complex should be in the .pdb file. Don't put pdbqt and different types of file extensions should not be used in the molecular dynamics. You should use protein ligand complex .pdb file. So once you import the structure, you can see how many chains are present, where the ligand has been binded, everything. You can visualize here. Once you go for ligand interactions, you can again visualize the docking interactions which it has. So now we'll be going for preparation of molecule, the first step. So here just by clicking this one, generate different hetero states with the pH. We no need to create that one because already in the docking it has been already done. So no need to create. Just we are minimizing the molecule and we are adding, if any there is missing of hydrogen or chains, we'll be adding that one in the preparation and we'll be doing the minimization. No need to create different with help of pH everything. We no need to do that one. So if you click the run, it will be the preparation will be going on in the so in the pre-process step, we'll be adding the side chains. For example, in the different softwares, as I explained, there will be in the protein ligand complex, there will be side chains will be missing. Side chains will be missing, or loops will be missing. So we can add from pre-process step, we can add the filling, we can fill the side chains, and from optimize hydrogen bond, their minimization takes place.
So GrowMax, like that and all, it's free, but Desmond is a software which is given as a package by Schrodinger. Schrodinger is a company where it is having different modules. In that, one of the module is Desmond, where this property is the filling missing residues and all, right, that will work based on the prime module of that thing. To add the missing residues, prime module will be used. So we don't have those things and all. If you try to run with the fill method, it will show the error. So we have to remove that and as well as the epic state.
Now the preparation of complex has been done. Now we'll be going for system builder. So already in the theory I have explained like different method, like we'll be adding periodic boundary conditions, we'll be adding water, ions, everything. Now we'll be building the model by using the tab called system builder. So here you can see we have water models which I explained, like single point contact, tip 3P, tip 4P, tip 5P, and we have co-solvents also, which if you want to work on the co-solvents for particular diseases, like in the alcohol reduction, everything, if you want to work, you can add, or in the formulations, if you use alcohol like solvents like that, you can use that one. For example, if you are doing, for example, if you are targeting TB and all, we have a different membranes in that. So we can select the particular lipid membrane by identifying that particular bacteria or viruses. We can select that particular lipid model, and then if you want to adjust to the particular atoms, if you want to, for example, if you want to target for GPCR, if it has any lipid membrane with particular amino acid numbers, you can select that one, you can adjust, and you can run the molecular dynamics for membrane protein also, but it is very computationally intensive. So generally for small molecules, we no need to set the membrane. Generally, we can add directly the water module. So then we'll be setting the boundary box. Periodic boundary box, which I explained, like we'll be restricting the atoms to the particular box. Here we can see different types like cubic, orthorhombic, hexagonal, different. But cubic, we can get more accurate result because it can replicate different, like which I explained, like box, everything, like in different all directions it will be there. So we can use cubic. So once if you click the show boundary box, it will boundary box will be visible to you. If you so then if you click the minimize volume, we are restricting the complex to the particular box. So that step is the preparation of periodic boundary box, which I explained. So after that we'll be ionizing. So ionization here, the algorithm is already capable of calculating by its own by using that particular complex. Here we'll be using sodium and chloride, which is main important in our body. So which we'll be using. So then if you run, the model has been built. In the jobs tab, you can visualize, or you can see at how much time it takes to do the particular job for the building of the model. So it shows once if you click that job tab, you can see whether it has been running or finished. After that, you can once you come back, you can see the box which we have the complex, restricted complex. We have solvated that one. We have added ions also. Now the system has become like we have built the system. So next we'll be going for simulation of particular model. So now we have another tab called molecular dynamics. If you click that one, so once you click the molecular dynamics tab, here you can see there is a tab called load. So we have built the model. To run the simulation, we need to load the model to the system. So once you load, it gives how much atoms for atoms wise, like for the complex ions and the water, how much it is present. We can calculate here. It already calculated and it gives how much atoms it is present. So here it is 1,39,249 atoms it is present. So the atom size is more. So then it takes more time to complete this particular simulation. So then here simulation time, as I already explained, based on your hypothesis and all, we can set the time. For example, if you are going for protein ligand complex, generally we'll be running for nanosecond scale. So here you can run not just for 100, you can run for 50, 60, 80 based on your complex and hypothesis. So then we have frames. So frames is like for each step we'll be considering that as a particular frame. So you can download the each frames and you can visualize at particular period or particular step how the complex is behaving in the simulation. You can download the each frames and you can see the you can visualize that particular frame. So then I explained the ensembles like NVT, NVE, NPT. Generally, in the molecular dynamics, we'll be using NPT because it is resembling close to the lab conditions and the results are very much accurate. We'll be using NPT ensembles. There are different types also. At last, we have NP80, NP gamma T, which we can use for these two ensembles. We'll be using for bacterial conditions. If you use, if you are targeting TB, that two ensembles we can use for that particular conditions. So after that, if you in the analysis, once the simulation done, we can visualize the analysis by clicking. If you click that particular run interaction analysis after the simulation completes. So once after we click this one, we can visualize the trajectories after running the simulation. So once we click that one, if you click the run, you can run the simulation. So in the Desmond, it is in the 4 to 5 clicks. If you go for Gromax, we have very much complicated, like each step we need to give a particular code, then only it runs. So it has lengthy procedure for Gromax, but in Desmond, it runs in with the 4 to 5 clicks. So this is the steps how we will do molecular dynamics in Desmond. So any questions in the steps? No questions? No. We can go for result analysis. No question. PPT. What PPT? PPT. 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 I have. PDF. PDF. Yes, PDF. So once after you run the molecular dynamic simulation, to analyze the results, we have another tab in that simulation interaction diagram. If you click that tab, after running the simulation, we'll be getting different files like out.cms, EAF, like different types of files we'll be getting. If you want to see the analysis of the particular simulation, if you load that .eaf file to the again to that particular software, we will be visualizing the results of particular that simulation. So once you load the .eaf file into the software, you will be getting a PDF with all the trajectories in the one PDF. So first you will be getting RMSD, that is root mean square deviation. Here you can see the graph. We have RMSD for the protein, we have RMSD for the ligand. So the blue color denotes the protein throughout the simulation, how it is behaving, whether it has any deviation from the original position, or that violet graph indicates protein ligand complex, which is aligned on the protein graph. So we have different options to here for we are running protein ligand. So we are aligning that for the protein. If you want to analyze secondary structure, or if you want to align that one on the loops, everything we can align the graphs and we can analyze this one. So here generally we are using as protein ligand complex. So we are aligning that protein ligand complex on the protein. So the generally the RMSD should be below 5 for better results. But if ligand RMSD is more than the protein RMSD, then the protein is not stable. So we need to optimize the ligand. We need to do docking again, and we need to run the dynamics again. Then we have RMSF, root mean square fluctuation. Here this graph shows for the protein. So the protein has different like alpha helix, beta sheet, everything. So in the graph, we'll be having the amino acid which has interaction for the particular ligand also. Though the atom which has interaction with the ligand will be having less fluctuation. If it has free secondary structure or loops, we'll be having more fluctuations here in the RMSF. So we can reduce, or we can reduce the loop by doing the loop modeling. We can reduce that loop and we'll be getting the less RMSF value. Protein secondary structure. So as I already explained, like protein has different like alpha helix, beta sheet. From this graphs, we can see like this blue color will be the alpha helix one, and this orange color will be the beta sheet one. At particular amino acid, which is alpha helix, which is beta sheet, you can visualize that one. You can understand from this graphs. Ligand RMSF. So while the simulation, like both will be in the motion. So at each this graph gives at each atom during the simulation which atom has more fluctuation. So to reduce this fluctuation, you can optimize this one. You can change the ligand structure, or you can optimize that one, and you can redock this one, and you can do the dynamics again. Protein ligand contacts. So while doing the docking, after doing docking, we'll be getting the results like which amino acid the ligand has interactions, everything. So here we will be having different types of interactions like non-bonded interactions, which I explained, like hydrogen bond, hydrophobic interactions, ionic interactions, water bridges, like hydrogen bond mediated with ionic bridges and hydrophobic. It will be like mixed. I generally will be considering the hydrogen bond as more strong when compared to this hydrophobic, ionic, and water bridges, because during the simulation, hydrogen bond will maximum it will be stable when compared to other bonds interactions. Protein ligand contact line. For example, if we saw the interactions which we have throughout the simulation for particular amino acid, which that color intensity, by the color intensity, you can get to know that which amino acid has more interaction. throughout the simulation. As the intensity increases throughout the simulation, it has retained the interaction. The color intensity, if it is less, meanwhile, it has broken the interaction and again it has regained the interaction. Ligand protein context, as we saw, we have different types of interaction we visualize, but from the molecule perspective, which it gives with which amino acid it has interaction throughout. For example, if you have run for 100 nanoseconds, it gives that percentage, for example, 67%, 69, 89. For example, if you have run for 100 nanoseconds, 89%. For 89 nanosecond, it is retaining the interaction. So from that percentage, you can analyze that, okay, for the protein ligand complex has run, the interaction has retained till 89 nanoseconds. So again, this graphs and all, it gives only the torsion profile of the ligand. So in this graphs, we cannot analyze exactly like what it is happening, because in it we don't have that package and all. If you to see the torsion profile, everything like a secondary structure like soluble axis of surface area, hydrogen bond analysis, everything. If you want to do, then we need to have package for that. In GROMACS, we can analyze SAS like solvent accessible surface area, like in the ligand, the structure, how much it is favorable in the water, everything we can analyze from that. In Desmond, we need a package for that. Till the ligand interactions, you can analyze torsion profiles. It gives like at which bond it has more torsion, or based on the color intensity, you can analyze which has less torsion, more torsion. To analyze this solvent accessible surface area, everything, we need a package for that. In GROMACS, you can analyze that one. These are the main trajectories which we will be getting through Desmond. Thank you. Any questions in the trajectory analysis or result analysis? Thank you. Yes.
We have one online question. Desmod free, what specification of laptop suitable to run this?
Laptops, generally we don't prefer to run on the laptops because we need more computational support to run the software. Gaming laptops also we cannot run that. So generally we have CPUs and GPUs. So generally in normal laptops, if you have like graphic card for around 4 GB, which is not sufficient to run the molecular dynamic simulation. So generally we need more computational support to run the like molecular dynamics.
So to add on to him, I'll be showing the system. Now just only 1 nanosecond we ran. So if you touch over here and you see that much amount of heat have been generated. So generation of heat will be there. This system architect is where here 2 fans, left side 2 fans, all the sides, all the fans were running. That much heat have been generated thrown out, even though the system is heated. But if you take the laptop, this laptop is also having a graphics card where we can able to run. But what the problem is, if more amount of heat is generated, the air circulation won't be there. The board, motherboard which we are having, right, all electricity board are there, right? Due to heat, it will melt and the system will blast or burst out or something else. So it won't give a long life run. So that and we'll never ever recommend this. Sometimes we'll open this window and we'll keep also, and it should run under AC. So that kind of sophisticated kit, this one.
We have one more question. For how much time we should do dynamics?
So based on your, for example, if you are generally developed a protein, if you generate a protein by using homology modeling, or just you want to run for particular protein, go for like around femtoseconds to picoseconds. If you are running for protein ligand complex, generally we prefer for nanoseconds, because both bonded interactions, non-bonded interactions, protein folding, unfolding, like loop modeling, everything we can analyze till nanoseconds. So generally for protein ligand complex, we'll be preferring for nanoseconds.
That nanosecond will also depend upon the atom size. As you showed, right, when the molecular build, the box size, after making the molecules build, what happens is number of atoms will be there. If in case some 10,000 atom, 20,000 atom, some protein and all will be 1 lakh, 2 lakh atom. So what happens, the system cannot able to take that much load. That time we have to reduce the time process and we have to work on. So it may varies accordingly. And nowadays, paper point of view, research publication point of view, what they are asking is either go for minimum 300 nanosecond. Previously, 1 nanosecond they accepted, then became 10, then became 20, 50, 100, now 300 and more. They are asking 300 more. Or certain times, what they are asking is they are asking the triplicate, as we are doing in the in vitro studies, right? The same way we are doing the triplicate values. So that condition is also there. Minimum time required is 200 at least. So that only after, for a longer run, you can able to come to know. And more based on this only, you can able to see whether the drug can be a covalent inhibitor or non-covalent inhibitor. That also is there. So as you have discussed about the annealing and other what do we call dynamics. Rather than that, one more dynamics is there, metadynamics, where you can able to come to know how much amount of energy or pressure is required to pull out the drug from the complex. Why? Because we know we have studied the enzyme classification, right? Competitive inhibition, non-competitive inhibition, and the same way reversible and irreversible. Reversible is the general mechanism which is happening. Whether the designed molecule which we try to bound with a protein is reversible in condition or irreversible in condition, we don't know. So to predict that also, we can able to work with a method called as metadynamics. So that it will give how much amount of energy will be required to pull out the drug or the ligand from the protein structure. So that is also required to work on.
In time perspective, for example, if you are running for around nanoseconds, as I already told in the requirements, you should divide the simulation into discrete time steps. For example, in nanosecond, you should run for some time, you should download the file, then again you should load. If you have computational power, you can run in one go. If you don't have the less computational power, run for some nanoseconds, then download the trajectory file. Then again, you can load that nanosecond where you have stopped, then you can run. So that simulation, it takes more time, but you can finish till which amount of nanosecond you can run.
Can you explain that RMSD and RMSF fluctuations? Because finally people can get it done from outside, but interpretation they need to know.
Back. So RMSD, as I already told, it is root mean square deviation. For example, during general steps, I explained like it will be in first, it will be in position 1, then it moves to the position 2. If it has more deviation from the original position, general moving is different, deviation is from the original position. If it has more deviation, then we'll be getting more like deviation graph in the RMSD graph. The RMSD graph, like intensity is less, the complex is more stable throughout the simulation. In the RMSF, you can see like if you have backbone, if you have loops and all, if it is free in the simulation, so it will be in the continuous motion. So we'll be getting more fluctuation throughout the simulation. If the loops and backbone, everything is stiff and it is not more extended with the ligand, so we'll be getting the less fluctuation in the RMSF graph. So in the interaction, for example, if it has particular interaction with the amino acid, it has less fluctuation throughout the simulation. This is what we can analyze through RMSD and RMSF.
So to add up to words, in general, as I said, right, someone asked about the docking studies. So yesterday, when if you are doing a docking study, there will be a term called as LB-RMSD, UB-RMSD. That means that drug, once it goes and bind with the receptor, it should not deviate anything. It should not disturb the condition. Example, if the switch is there on and off, in the sense, I have to go touch politely, switch off, switch on. That means there is no disturbance towards me or towards the switch. That's the condition. If I go and hit a forcefully, either the switch may get damaged or I may get damaged. This is the concept over here, RMSD. So from the RMSD, what we can able to say is once drug goes and binds with the receptor, it is not disturbing its original nature, so that the RMSD will never be deviated. The word itself, deviation. So this is the result of whole protein nature. So next graph, what I showed is RMSF. Root means square fluctuation. So the protein, I said, individual amino acids should be the right. The whole body I have discussed. If someone come and try to hit me, I'm holding for a while. So something should not damage to me, right? Either my hand is damaged or not. Individual amino acids I'm speaking. That is the root means square fluctuation. So rather than that, if you want to able to work on another term called as RG, radius of gyration, which we can't able to perform in this Desmond, but we can able to perform it using GROMAX. RG in the sense individual atoms. So everything can be calculated once the drug binds to the receptor during the simulation time period, the whole protein is damaged or not, or individual amino acid is damaged or not, or else individual atom. So as a chemist point of view, finally it ends with the atom. So the motion of an atom will be predicted through the dynamic studies. So that's the purpose. And rather than that, one more study will be performing SASA, solvent accessible surface area, when the drug bind, the protein nature will be slowly shrinked, so that it won't have the contact of the outside. That also can be predicted using GROMAX. But here, this is the results where we can able to get down from the things 3.2, 3.2, and you, with a graph point of view in a software, if you go and check, you can able to see exactly what is the thing, what is the value and all. So the results output you can able to show. And down, down. Stop. So now the latest, the image which you have showed is the older version. Now in the newer version, we can able to see the green color border, right? This green color border is the where the ligand is interacting, to which amino acid is interacting. Either it may be hydrogen bond interaction, hydrophobic interaction, pi-pi stacking, whatever the thing may be, but it don't bother. But a type of interaction. During that interaction time of the ligand, is there any fluctuation? Now by seeing the results, there is no fluctuation. So the results are fine. So next. So this secondary structure and protein structure elucidation, when it will be utilized in the sense as Mr. Basamira have discussed about homology modeling, right? Based on their studies, it can be done. So based on that, it can be what we call, after his study, he have designed a protein. So it has to be validated. It has to be validated. For that time of period, you can able to see how much percentage of helix is there, strand is there, secondary structural elucidation is there, that we can able to predict. So most probably for the docking point of view or the this one, we won't much use. Scroll down. Yes, 1 minute. So not only for the protein structure, even for the ligand structure also, we can able to calculate the root mean square fluctuation, whether the ligand is fluctuated or not. This indirectly, the results will be reflected in the torsion, the last slide. That will be there. Next. Yes. So this is the protein, what do we call, what type of interaction, even the compound may be present as during the motion in docking and all we have seen, right? The results will be showing only one type of interaction, either hydrogen bond interaction, pi-pi stacking, or whatever the thing may be. But during the period of dynamic simulation, you can able to see three types of variation. What are the three types of variation? It have attracted as a hydrogen bond interaction, hydrophobic interaction, as well as water bridges also formed. But when you are doing docking, we will never do the water bridges. Water we will never consider. But body will take water mediated interaction. We know only the hydrogen bond interaction, right? But here, this is water mediated interaction have been created. Next. Yes. So this is the protein ligand contact, like a barcode, how far, how intensely it have been interacted, either in any form, either the, as I showed in the previous one, right, how far it has been interacted. So here you can able to see ISP in these two are more, and TRP 31 is fine to be interacted. So these are the key amino acids interacted with this particular protein, particular ligand. Next. So little minimize structure. Yes, little down. So now you can able to see there is a 92% of interaction where it is water mediated interaction. It is not a regular interaction. So here you can see 73%. If example, I have run a 100 nanosecond period, in that 73, 73 percentage time, it have been interacted. 73 nanosecond, it have been interacted. So the same way here, what type of interactions we want, we have got. This is the ligand protein contact. Previously protein ligand contact, how protein is interacting, and now how the ligand is interacting with the protein. So this is the, we'll be studying our pharmacology, right? Pharmacodynamics, what the drug does to the body, what the body does to the drug. Same mechanism, what protein does to the ligand, what ligand does to the protein. So in which way they are interacting, either through phone or WhatsApp or message, such kind of communications. They are the different type of bonding. So these are the things, and this will be based on the, what we call, wait. So this green color, you can able to see this is one. So as I said in the general the theory sessions, always we need we have to look for the 3D structures only. Why? Because our bodies of a 3D nature, where the hydrogen dihedral angle, torsion angle will be playing a major role. The flexibility will play a major role. Sometimes some of the drug you might have seen only with the single bond. Sometimes some of the drug you might have seen with the triple bonds also. Saturation, super saturation point, right? So that it is required. Based on this, in case if example there is a functional group, only CH3 is there and maybe interact in the sense, I can change the group as a double bond or like a CH2, and again I can able to retry as a thing. For a lead optimization process, here this values will be utilized, which is more, which is less. Where it is falling, based on that we can able to say this region has to be adjusted or modified. So this how will be what do we call, the same way as I said, right? RMSD, radius of gyration. So only for ligand properties, only with the ligand properties they have given. But in GrowMax, if you run, you can able to get the RG, SASA in GrowMax for proteins. But here the advantages, it can able to run the RSD for both protein and ligand together. But if you run in GrowMax, it will run separately. If I want to compare in the sense, I should run a protein separately and I should run a ligand separately. So this is the advantages and disadvantages of the things. Any other questions?
So, sir, like for the protein ligand contacts, if there is interaction with a water bridge also, so then how should we report? Because if it's the normal hydrogen bonding, then we'll tell with alanine 324, a hydrogen bonding is there for 89%. This proves that stability. In case if it is with water bridges or like ionic and hydrogen, then how should we report?
Generally, like as in the question answer, we have answer, like for example, we have hydrogen bond interaction, ionic bond will be reporting like, but here we'll be generally telling like it has water mediated interaction. So water mediated interaction has less affinity when compared to hydrogen bond, everything.
Okay, so
We
full
can report
hydrogen,
it. We can report it that in the paper.
but the complete hydrogen bonding will be the most.
Most stable one and most affinity it has.
Okay, sir, thank you.
Any other questions? ಇದನ್ನು ಹೋಗುತ್ತಾ ಜಾಬ್ ಪ್ರೊಫೈಲ್ ಹೋಗಿ ಸ್ಪೈಸ್ ಯಾವ್ದಾಗಿದೆ ಜಾಬ್ 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 ಫೋಟೋ ಜಾಬ್ 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 once after you run the simulations, we'll be getting different file formats for as I already told, if you want to divide the total simulation time into discrete times, and if you want to run, we'll be using out.cms file where you can load that file at last point which you have stopped the simulation. You can continue from there. So out.cms file will be using to continue the simulation. We'll be getting CPT files, CPT and EAF files, where we'll be loading to get the simulation interaction diagram, the trajectories which run throughout the simulation. And we have another log file also. For example, in the log file, we'll be getting throughout the simulation. For example, if you are run for 50 nanoseconds, what are the steps it has been taken, where it has been deviated, everything we can see in the log file. So these are the main general files which we'll be considering in the output file of Desmond. So if you want to visualize this, if you want to visualize this one, for example, PPT in the ionization slide. PPT. PPT. Ionization. Mother, click. Full slide. Full slide. Close and open one. Full slide. See, for example, if you want to visualize the simulation, like how it has been, how protein and ligand is behaving throughout the simulation, you can see this file, the video. It will be as it is, like it will be moving. The ions will be, the water will be moving with the protein ligand atoms. So if you give out.cms file in the Desmond software, if you click, there will be an option called trajectories. If you click that one, you can analyze at which nanosecond or which frame how the protein ligand is behaving, how atoms is moving, everything you can visualize the complex, the simulation through which you have run. We can not only see the results, we can visualize how it is behaving throughout the simulation. That also we have here.
Any more queries?
Thank you so much.
Thank you to the session speaker for providing in detail information on the topic. Now I request session chair Dr. Vijaya Banu to honor our session speaker.